For your specification, you need to be aware of how microscopy techniques have developed over time. The light microscope was first invented by Zacharias Janssen and his son Hans in 1590, and in 1609 Galileo began work on creating a better microscope. Since then, the light microscope has developed massively, and we now have light microscopes that can magnify up to 2,000 times. But most, if not all, of the microscopes you will come into contact with will only magnify several hundred times. In 1930, the electron microscope was invented by Max Noll and Ernst Ruska. These microscopes can magnify up to 2 million times, so allow biologists to see and understand more about the subcellular structures inside cells. There are two main types of electron microscopes: transmission electron microscopes, which give 2D images with very high magnification and resolution, and scanning electron microscopes, which give 3D images with lower magnifications. Magnification is defined as the ability to make small objects seem larger, while resolution is the ability to distinguish between two separate points. The resolving power of a microscope determines how much detail can be shown in the image that is produced. There are several differences between light and electron microscopes that you need to be aware of. Light microscopes work by using a beam of light to form an image, which can be viewed directly, whilst electron microscopes work by using a beam of electrons to form an image. Electron microscopes are larger, heavier, and more expensive than light microscopes, and have much higher magnification and resolving power. But they cannot be used to view living specimens, as these microscopes require there to be a vacuum in the tube. Therefore, when viewing things like live cells, light microscopes must be used. So, for your course, you need to be able to do a couple of calculations involving microscopes. Firstly, you should be able to calculate the magnification you are using with a light microscope. To do this, you need to multiply the magnification of the eyepiece lens by your objective lens. For example, if you're looking at a cell using the 40 times objective lens, you will multiply this by the power of the eyepiece, which is usually 10 times. Therefore, your magnification is 400 times. Secondly, you need to be able to calculate the size of an object viewed through a microscope. The easiest way to do this is to use the magnification triangle. I remember this by thinking I am, like the I Am's cat advert. On the top of the triangle, you have the image size, and on the bottom of the triangle, you have actual size, the size of the real object, times magnification. To find the magnification using this method, take the size of the image divided by the size of the real object. And to find the size of the real object, take the size of the image divided by the magnification. For example, if you're looking at an animal cell that measures 2 mm across and the magnification is times 40, the actual size of the cell will be 2 divided by 40, which is equal to 0.05 mm or 50 micrometers. One of the required practicals on your specification involves observing plant and animal cells under a microscope. You're most likely going to be looking at onion cells and/or pondweed cells, the scientific name Elodia. You will use tweezers to remove the inner layer of cells from a piece of onion. This layer will then be placed flat on a microscope slide with a drop of iodine and a cover slip placed over it. Be careful when you're placing the cover slip, as you don't want air bubbles to form. First, look at the cells using the lowest magnification on your microscope, and then increase the magnification until you can clearly see the cell structure. As onion cells are plant cells, you should be able to see the cell wall, cytoplasm, and vacuole. Then, repeat this process using a pondweed leaf, although you'll be adding a drop of water this time instead of iodine. As well as the structures visible in the onion cell, you should also be able to see chloroplasts, as these are photosynthesizing cells. Draw a diagram of each cell, making sure to label the structures that you can see. Record the magnification you've used to observe the cells, and then you can use this to work out the actual size of the cell using the above magnification triangle.